Okay, so this will be the recording for the lab on tissues. Okay, part of this is a review, but I think it's important that we provide that review, and we're going to look at 6 of the tissues that you have. So you need your tissues lab exercise. You have a place to put a definition of the word tissues, a group of similar cells and cell products with a distinct shape and functions. Sometimes it's the similar cells that are the big visual cue in the tissue, and sometimes it's the cell products, depending on the type of tissue. Histology is the study of tissues. So when you have a biopsy, when you have blood work, when they remove a body part and they send it to pathology, that's a histology report, histological report. 4 major types of tissue. You have a table in chapter 5 that covers this. Epithelial tissues cover internal and external surfaces. So your skin on your cheek and the inner lining of your cheek. Internal, external surfaces. The outside of your nasal passages and the internal surface. It affects absorption. If it's multiple rows of cells, like we see in this example, over here, oops, it goes from here out to here. That's going to reduce absorption or prevent absorption. If it was 1 row thick, it would enhance or facilitate absorption. And secretion. The glandular tissue in the body is all going to be epithelial tissue. So your salivary glands, your endocrine glands, the mucous membranes, serous membranes, those are all epithelial tissue. The connective tissues are going to anchor things together or bind structures together. They act as a cushion, meaning that they create a barrier or a cover on something. They are going to absorb trauma, physical shock, act as an insulation, keeping things separate from each other, or to provide a cover and provide a place to allow immune activities. So here we have one kind of connective tissue. Here we have another, and this one is the same as what we see over in this picture. So, in between things. Nervous tissue detects stimuli, which is called irritability. Conduct impulses or excitability, and store memory. Now, without getting into too much, the storage of memory is not like on a flash drive or on a computer or in the cloud where there's a physical location. With us, storing memory is a pathway, and it's using that pathway repeatedly that creates memories. Muscles contract and relax. Instead of contract, you can use the word shorten. Instead of relax, you can use the word lengthen. They're also excitable, so they also conduct electrical impulses. Perspectives on tissues. Here you've got a hard-boiled egg. These bottom three cuts give you all the information that you need. The upper two cuts that we see here and here, or the outer two cuts, don't give you the full picture. Oops. It just shows you one kind of material. Doesn't show you the true shape. Also, keep in mind that the cut went in this direction, but they rotated it so that you could see into the subject or into the specimen. This middle cut's the only one that gets you the true picture of the elbow macaroni. You have some hollow areas and you have the solid portions. If you didn't see the whole thing, you wouldn't know that these two circles are part of the same structure. So again, the cut was made in this direction. Oops. This direction, and then they rotated it so that you could see into the specimen. Over here, you have the duct of a gland. You've got a longitudinal section over here. And if you didn't, again, see the whole thing, you wouldn't know that all of those are pieces of the same tube. So you have to keep that in mind. Here they sliced it lengthwise and then removed the top piece. So in tissues, you need to look at the captioning, where you've got longitudinal sections where they cut the length, cross sections where they cut it in half or lengthwise, and then we use the term oblique for diagonal. Now, we're not going to see any oblique sections. However, we will see some oblique versions of hair follicles. When they take a specimen of skin, they cut straight down. However, hair follicles sit at an angle. And so you're going to get an oblique version of some of those hair follicles. And that's because the cut was made straight, and the structure was at an angle, not the fact that they made the cut at an angle. Make sure you look at the captions of the tissues slides, and if you were to take a slide and put it on a microscope, then you would actually have to look at that and see what kind of section you have that you're going to look at, so you know what kind of view you're going to have. Okay, with regards to epithelial tissue, one of the words in the name of epithelial tissue is called the cell arrangement. So these are three examples of cell arrangement in epithelial tissue. Simple means you have one row of cells. Stratified means you have multiple rows of cells. Now, this is their version of pseudostratified, which I don't like. Oops, okay. So, with a pseudostratified, Oh, I lost my pointer. There we go. Okay. You're going to have one row of cells because they all touch the basement membrane. But what they don't do is all reach the surface, the free surface. Their nuclei are staggered, which is why it looks like they're multiple rows So on epithelial tissue, this is the basement membrane where they're attached. This is the free surface where they're not attached, or the unattached side. The second word in the name of an epithelial tissue has to do with the shape of cells. Squamous refers to, or squamous is a squashed, flat, irregular shaped cell. The nuclei is even flattened. Cuboidal, they're about as tall as they are wide. The big thing is this round, centrally located nucleus. Sometimes that's what you're going to see and not the cell membranes. Columnar, they're long and tall. They have a large oval nucleus. Here's a true picture of pseudostratified columnar epithelium. And this is to illustrate goblet cells. The goblet cell is wide at one end and narrow at the other, and typically has, this is the cilia, typically has a large glob of mucus in it. Another version of them would be back up here in my drawing, where we can see wide at one end, narrow at the other. Again, it can be wide at the bottom or at the basement membrane and narrow at the top. But that's a goblet cell. It's a modified columnar cell, like a wine goblet. Then you have the word epithelium. So 3 words in the name, at least. Simple, squamous epithelium. Stratified columnar epithelium. Sometimes they'll include things like ciliated, pseudostratified columnar epithelium. Okay. 3 traits of epithelial tissue. One surface is attached to a basement membrane, and that would be along here. One side's unattached, out here at the lumen. Sometimes you have 2 basement membranes next to each other. So you've got a long extension of that tissue, and then here you have a tube, and the 2 basement membranes are right next to each other, with the lumen in here where it's the free surface. No direct contact with the blood supply. This tissue depends on other tissues passing it nutrients and carrying away the waste products. This tissue, because it's on surfaces and because it's carrying out secretion a lot, continues to carry out cell division even after reaching full maturity. A lot of the tissues, their cells lose that ability once they reach full maturity. So they can't always replace themselves. So because it's on surfaces, they need to replace themselves. If you think about your skin, the epidermis that you have today is not the epidermis you had several weeks ago. You get a completely new set of cells every couple of, I think it's like every 6 weeks. Okay. In connective tissue, the big thing is the matrix. Extracellular material produced by the cells of connective tissue, composed of both ground substance and protein fibers. The cells make the ground substance and the protein fibers. We use the word fiber differently depending on what kind of tissue that we're talking about. Here, we're going to talk about protein molecules. Either way, a fiber is something long and narrow. In this tissue, you have these really narrow extensions, long, black, dark, and then you also have these wide pink bands. So I can get over, there we go. Wide pink bands. Those are two different kinds of fibers. The white area is the ground substance. Over here in this blue one, the bluish material here in the middle is a mixture of the protein fibers and the ground substance. So that way you'll just refer to it as matrix. The bubbles are the cells. In the pink one, you can see the nuclei of the cells, but you can't see the cell membrane. Okay, ground substance is a solution with 4 substances: water, glucose, aminoglycans. We break it into 3 pieces: glucose, aminoglycans, proteoglycans, and glycoproteins. These are conjugated molecules. They're part carbohydrate and part protein. Here, you can see the different consistencies. This one has a solid ground substance. This one has a semi-solid. And over here, we have a liquid. So the consistency of ground substance can range from a liquid to a solid. 3 kinds of protein fibers. Collagen fibers are what we see over here. Wide, thick bands that provide tensile strength. Reticular or coarse fibers, which is what we see here in this purple picture. They have uneven surfaces. They have different thicknesses. So they're wide in some places and narrow in other, also called coarse. And then down here, these narrow lines are elastic fibers. They provide flexibility. Sometimes there's 1 kind of fiber, sometimes there may be 2 kinds of fibers. If that's their main physical, visual attribute, then we call them fibrous tissues. So, specific cells found in areolar tissue. Now, keep in mind, you are not going to be able to see the difference between one and the other. The fibroblast is a cell that makes protein fibers. Macrophages are large phagocytes, and leukocytes are cells that we find in areolar tissue, reticular tissue, and blood. So several different kinds of cells in areolar tissue. Again, all you're going to be able to see is the nucleus. And that's these dark spheres that you see here. Keep in mind that this is the one that has two kinds of protein fibers and ground substance that you'll need to label on your drawings. Reticular tissue has reticulocytes, which are special fibroblasts, macrophages, and leukocytes. Again, all you're going to see is the nuclei of the different cells. Dense tissue has just fibroblasts. So if we look in this, these dark hyphen looking things are fibroblast nuclei. The waves are the collagen fibers. The white would be the ground substance. This tissue, dense regular, has a organization to the collagen fibers. This tissue is also called white tissue because, like in that picture of the protein fibers with the white collagen fibers, it's white inside of people and other mammals. Dense tissue also comes in irregular, and that's what we'll see in the dermis, where there's no organization to the collagen fibers. So all this pink, rather than being nice wavy bands, is all wadded up. Yes, there are still fibroblasts. It's in order to make spaces for the accessory structures of integumentary system and other blood vessels and other things in that tissue. So why is blood a type of connective tissue? Because it has matrix. Now, here, the pink and the purple that you see are the formed elements in blood. The white is the ground substance. The only time that you're going to see protein fibers is during coagulation. So that's why it doesn't look like you've got protein fibers, because they haven't been assembled yet. So yes, blood is connective tissue, and we have an entire chapter that we'll cover and look at blood, so we won't be looking at it here. Muscle tissues. Here we use the word fiber to indicate a long, narrow cell. Muscle fiber, muscle cell, same thing. Some muscle fibers have striations. These alternating dark and light bands are called striations. In the other semester, you should have learned why we have these in some of the types of muscles, and that's because of the organization of the protein filaments inside the muscle tissue. Intercalated discs are only found in cardiac muscle tissue. So that would be this example over here, not that. They are going to look like darker versions of striations. Oops, there we go. They connect two different cardiac fibers together. So intercalated discs are a way for cardiac fibers to be anchored to each other. And that's so when they contract and relax, they get shorter, they get longer, they don't pull apart. So the other fiber would be here, and this is kind of like an extra thick seam. And so when the cells contract and relax, they don't pull apart. They stay together. Skeletal muscle, you have a table to fill in. The attributes of skeletal muscle, yes, striations are present. Many nuclei per fiber. You can see several nuclei for this cell. You can see lots of nuclei for this fiber. The shape, they're long parallel cylinders, and they're under voluntary neural control. You have conscious control of your skeletal muscle tissue. You send out conscious messages to tell them to contract and to relax. Cardiac muscle, yes, striations are present. One nucleus per fiber. They're short, branched cells that run end to end. All of that is the shape. The branching, you can see here, here, here. These are the myofibrils. They're under involuntary neural control. Your autonomic nervous system controls the cardiac muscle tissue. Smooth muscle tissue, no striations, one nucleus per fiber. The shape is called fusiform. It is wide in the middle and narrow at each end. Or tapered at each end. It's under involuntary neural control. Smooth muscle tissue is also called visceral muscle tissue. All right, so on the boxed version of your handout, the drawings portion, you need to draw simple cuboidal epithelium. The simple cuboidal epithelium, this is one row. This would be the lumen. You may have to zoom in to that so that you can actually see the tissue. Simple columnar epithelium. This would be one row. This would be another. So you need to draw examples of them on your paper. In your textbook, you'll also find tables that have representative functions and locations. Now, keep in mind now and throughout the rest of the semester, I will always count the word protection, protects, wrong. Protection and protects is too vague. I want to know what it does. Support, you better have a word in front of it, a descriptive term. Is it nutritional support? Is it physical support? Is it immunological support? So make sure that you do it in short phrases. So secretion, absorption, produces mucus, things like that. Locations. Lying underneath connective tissue is too vague. I want to know where it's found. You need 3 of those unless they only give you 2, 1 or 2. So you'll need to use your book for that portion of your handout. Areolar tissue, one of the 2 connective tissues. Again, you may have to blow it up bigger with areolar tissue. Oh, something else. In each of the different types of tissue, major types of tissue, you have certain things that you have to label on the drawing. I will count that wrong if, count points off, if you don't do the labeling. So make sure each section, the epithelial tissues require certain labels. The connective tissues require different labels. The muscle tissues require another labeling. So these are the pictures that you need to get familiar with. With your drawing, you're focusing on the different attributes so that you can be tested over them. Dense tissue, another connective tissue. Again, you may have to blow this up so it is something you can draw. Keep in mind that dense tissue and smooth muscle tissue are not the same. The fibroblast nuclei look like little hyphens. The nucleus of the smooth muscle tissue is much bulkier. Cardiac muscle tissue, you're looking for the intercalated discs scattered, and they're going to be parallel to the striations. Now, of course, there's only one location for heart, cardiac muscle tissue, and that would be the heart. So don't start inventing locations. If your book only gives you 1 or 2, that's all you need. If it gives you a bunch, then 3 is the magic number. So those are your 6 drawings that you need to complete for the tissue lab.